Do you remember the swine flu outbreaks in 2009? The uncertainty and fear of the spontaneous pandemic? The white masks millions wore for protection? Schools closing down? By the beginning of the outbreak, the World Health Organization counted 18,500 deaths confirmed by lab tests. This unexpected pandemic took America by storm and struck its youngest generation first. On April 15th, a 10-year-old patient was being tested for influenza in a California lab as part of a study when the CDC discovered that the virus detected was new to humans. It was only 2 days later that another infection with the same virus was detected in an 8-year-old, just 130 miles away without any known connection to the previous patient. These influenza viruses were different from any other kind seen in either humans or animals. These influenza viruses were different from so we thought. As the CDC began to investigate, there were 12 cases that were discovered to randomly occur with North American lineage to swine influenza viruses during December 2005 to January 2009. 11 victims had some type of interaction, 5 direct and 6 proximal with pigs, while one case's origin was unknown. There was major concern regarding the 2 cases that occurred in April that had no possible connection to each other, leaving the remaining conclusion to be that the H1N1 virus had managed its way into the human population and was currently rampant. On April 18, 2009, the U.S. International Health Regulations Program reported the influenza cases to the World Health Organization.
There is the basic idea that cells form a new subtype through combinations. Reassortment is the mixing of genetic material into a different combination. This is particularly common when 2 similar viruses infect the same cell and exchange genetic material. The viruses inject their genetic material into the host cell. Take a look at the different green and orange segments located inside the host cell. They will combine to make a new strain that has properties from both its parent lineages. This process is responsible for the H1N1 outbreak as swine, avian, and human influenza genetic sequences combined. Antigenic shift is a specific case of reassortment that addresses the mixture of surface antigens of the 2 original strains to create a new form of a virus that has different surface identification.